Okay, so we're on 7.4. Let's get 7.3. 7.4, human genetics and pedigrees. So we're going to use a combination of methods to study human genetics. And we'll go through a few of them today on how we study how genes are passed on to children, how we can predict the chance of having a certain trait. Ben. Oh, sure. Oh, sorry, you have to carry that. Yes. That's nice. It's just like 3 hours, so it's like, yeah. So we had this slide down? No. Okay. So we have a wide range of traits in the human population because we have a wide range of genes that control those traits. And most traits you can see in a person are from a wide range of genes working together to create a wide range of traits. So it gets quite complex when we look at humans. So it's not just a simple one single gene that gives you a certain trait. But even though humans are very complex, genetics do follow a similar pattern that we see in other animals and other organisms. So we can look at plants and animals and see patterns in them. And it's similar to the patterns we see in humans and the genes that we see and how they're passed on. So the basic principles of genetics are the same in all sexually reproducing species. So whether you're looking at a plant, an animal, or a human, they all need male and female to create a new organism. So plants have male and female parts, so do animals, humans. So because of that, we have similar properties or similar principles that we can apply to all those organisms. But the inheritance of human traits many times is quite complex. And a lot of times we don't even understand how we get certain traits. We've discovered a lot of genes in the human species, 30,000 genes about at this point. But we only scratch the surface on the genes that are there in the person and how they work. There are some single gene traits, and those are important in understanding human genetics. So one single gene trait would be blood type. We can look at one gene that controls blood type. But genes for hair color, eye color, those are several genes. And so it's not a single gene for those traits. And most traits are that way in a human. Lots of times diseases are single gene traits. And so most of our knowledge about human genetics are from diseases. Because it's usually due to just one gene that's damaged that creates a disease. So we talked about this before, that females will carry sex-linked genetic disorders. So females have 2 X chromosomes. And because of that, they can carry a damaged gene, but not have it themselves. But for males, they only have 1 X chromosome. So if there's a gene that's damaged on the X chromosome, they will have that disorder. The expression of the disorder depends on which parent carries the allele in the sex of the child. So whether the mother or the father carries the gene and whether the child is male or female will affect if they have the disorder or not. So in the X chromosome, there are roughly 2,000 genes that they've discovered. And this just shows you a few of the genes that they know about. And so there's a gene for color blindness right here. There's a gene for hemophilia, another gene for color blindness down here. So there's genes for deafness as well. This chromosome controls a lot of different traits in the body. It's a lot larger because of that than the Y chromosome. The Y chromosome really only just gives you male characteristics. So we have these down. So color blindness doesn't usually mean that you can see no color. Usually if you're colorblind, you can see some colors, just not a wide range of colors. And so this shows you, on the left-hand side, the true image. On the right-hand side, a person who's colorblind, what they would see. So they would just see mostly what looks like brown. They might see blues, but many times they don't see reds. Reds are not visible to them. So red-green color blindness is the most common form of color blindness, where they can't distinguish between reds and greens. To them, it looks the same. So it affects 1 in 25 people. There's a test you can do to see if you're colorblind. So if you can see the numbers in each one of those, then you're not colorblind. But if there's any ones you can't see the number, then you have a colorblind, at least one colorblind quality. So again, being colorblind means that you can see some color, just you can't see a wide range of colors. You can't tell apart certain colors. And so if you can't see the 7 or the 12, then you have red-green color blindness. You can't see reds and greens apart from each other. So it's a lot more common in boys than girls. It affects 1 in 12 boys, 8% of them, but only affects 5 out of 1,000 girls. 0.5% of girls are affected by this. So because it's a lot more common in boys than girls, then we know it's sex-linked. We know it's on the X chromosome. So we're going to look at what a pedigree is. A pedigree is like a family tree, but it's a family tree that focuses on certain genes that are passed on. So it's a chart for tracing genes in a family. A pedigree keeps track of certain phenotypes, things you can see in a person. And by tracking them over generations, you can infer what genes the parents and grandparents and children have. So we're going to look today at pedigree, which is based off one phenotype. It's hairline, whether you have a widow's peak or a straight hairline. So if it's a capital W, it's widow's peak, which is what you see right here. So if you were to push your hair back and you see this indentation right here, kind of like a heart shape, that's a widow's peak. So girls and boys can have this. It's not sex-linked, it's autosomal. So it's not on the X. So you're going to see the same amount of boys and girls having this trait. Lowercase w is a straight hairline. And so that would just be, if you just were to have no indentation at all, it's just straight across. It's called widow's peak because at one time they thought that if you had this, then you would survive your husband. It's just a legend that they had. That's why they call it that. Yeah, like be live longer than he does. Yeah. Because if you're a widow, then you lost your spouse. Yeah, I don't know. It's a good question. I don't know what they would have thought about that. I'm not sure. So looking at this pedigree right here, go ahead and write this down. You know, this box right here and the line and the circle, and we'll talk about what this means. So these parents right here, the square represents a boy or a male, and then circle represents a female. So make a note of that too. Make a note in your on your notebook that squares are males and circles are females. And then a bar in between them represents a match, like where this is a couple. And so this would be parents. They would. And then you have circles that are you have ones that are filled in or ones that are not filled in. So the ones that are filled in represent a trait, and the ones that are not filled in mean a different trait. So in this case, if it's not filled in, it's a straight hairline. 2 lowercase w's give you a straight hairline. A filled in circle or square means they have a widow's peak. Let's look at the children. So now you see this line that comes down the bar, and the ones that are connected right here are their children. So this couple has 3 children. And then right here, this would be the spouse of that child. And this is the spouse of that child right there. So how many girls did this couple have? 2, and then 1 boy. So 2 girls right here. Circles are girls, and then 1 boy. What would this one look like? Widow's peak or not? Yeah, widow's peak. And this one, straight hairline. And that's a boy with a widow's peak. So blue ones mean widow's peak. White ones mean no widow's peak and straight hairline. So we know if there is no widow's peak, they have to have 2 recessive genes for that to be the case. But for somebody who has a widow's peak, there's 2 possibilities. They could be heterozygous and carry the gene for a straight hairline, or they could be homozygous. Well, the kids in this family right here, we know for sure what they are. We know for sure that these must be heterozygous. Because this parent right here gives one of the 2 genes to their children. Well, this parent, he only has a lowercase w to give to their kids. So they all inherited a lowercase w from the dad. The mom could give either uppercase or lowercase w. And so if she gave uppercase w, it was a widow's peak. But this one, lowercase w from the mom and from the dad. But this right here is outside the family. We don't know anything about her genotype. We know she has a widow's peak, so we know there's 1 capital W. We don't know if she inherited another capital W or a lowercase w. We don't see her parents, so we don't know. But now we can see, so the F1 generation, F2 generation, so grandparents, kids, grandchildren.
We have to keep writing that down.
Yeah, it'd be good for you to know, understand this. So yeah, you should draw this all out. And then this couple right here has 3 kids. And also 3 kids for this couple right here. So what can you tell about here? So we know, again, that is the same as right here, that these kids must have inherited a lowercase w from the dad because the dad only had lowercase w's to give to the kids. The mom had uppercase and lowercase w's to give, and so some had uppercase W, widow's peak, others had lowercase w, which was a straight hairline. But for this other couple over here, we really can't tell about the mom over here if she had uppercase W or lowercase w as her other gene. If one of the kids had a straight hairline, though, like we see over here, then we would know that the mom had a lowercase w. But because we don't see any kids with a straight hairline, we're not for sure if she carries a gene for straight hair or not. What we can also think about too is whether it's autosomal or sex-linked. So we would count up how many boys have this trait, how many girls have this trait. If there's a different number, like a huge difference in number, then we would think it's sex-linked. So we can do that. We can count how many girls have this trait, how many boys. So for widow's peak, we could count up. So we have 1, 2, 3, 4, 5, 6 girls who have it. How many boys have it? We have 1, 2, 3. So because there aren't a lot more boys than girls who have this trait, it's autosomal. There are certain traits today that we can see that we haven't seen a gene for, but we assume are sex-linked because there's a lot more men than women who have that trait. Autism is an example of that. For autism, we don't have a gene for it, but we think it's sex-linked because there's a lot more boys who have it than girls. And so they suspect that it is on the X chromosome. They just haven't found a link yet to that. But any traits that we see that are more male dominant than female dominant, we assume are sex-linked and on the X chromosome. So we're going to look now at a different kind of pedigree. This one is for a sex-linked trait. So as I mentioned, if you see a phenotype that's more common in males, then the gene is likely to be sex-linked. So we're going to use another pedigree. It's going to be similar to what we did last time, where we have squares for males, circles for females. The difference would be the shading. What we do is to show that there's a carrier, somebody who carries a recessive gene, we're going to have a half shaded circle for those who carry a trait, but they don't express that trait. So this is for color blindness. So if you see a lowercase m, just one for the male, they're colorblind. For a female, she has to have 2 lowercase m's to be colorblind. So this female carries a gene for color blindness. She can give it to her children, but she's not colorblind herself. So looking at the children, so this couple has these 4 children right here. And what do we see? So the dad can only give a capital M to the children. So each child will get a capital M from the dad. That's for the girls. Now, if they're males, though, they get a Y instead. So for a male, he'll get a Y from the dad, and then for the mom, he could get either capital M or lowercase m. And so half the males would be colorblind because half of them would get lowercase m from mom, and then they all would get Y from dad. But none of the females would be, because all the girls will get a capital M from dad, in which case they will not be colorblind. So we can see down here that for these circles right here, some of them for the females are carriers. Some have a lowercase m. Some just have a capital M, and they're not carriers. But for the boys, what do we see? Well, in this case, both the boys are colorblind. So they both got lowercase m from mom and a y from dad. Some of the boys could have not been colorblind. They could have got capital M from mom. But in this situation, this family, all the boys are colorblind. And then for the girls, we don't know about this girl right here because she wasn't married and we don't see her children. So we're not for sure if she is a carrier or not. But for this girl over here, because she was married and had a family with colorblind kids, we know that she must have carried the trait of colorblindness. So carriers are half shaded ones where we know for sure that they have a capital and lowercase m. So on a Punnett square, we don't always know for sure the genotypes. We have to guess sometimes. So there are sometimes when we're not for sure. The dominant trait that a person has, we don't know if they're homozygous or heterozygous for that trait until they have kids. And then if they have kids who have the recessive trait, then we know, well, they must have carried the recessive trait. So let's count how many boys versus girls have this trait. So you count the filled squares. Those would be how many people have that colorblind trait. So you can see that on here, only the boys are filled. That means that only the boys are colorblind. None of the girls have colorblind in this family. So 4 of the girls are carriers for colorblindness, but none of the girls are colorblind in the family. And so because of that, because we have a lot more males than females who have this trait, we know it's sex-linked. Did we talk about karyotypes before this year? I forgot if we brought that up or not. Oops, sorry, I'm not sure what happened there. There we go. Karyotypes, basically what you see is all the chromosomes in your cells arranged in order from longest to shortest. So number 1 is the longest chromosome. 22 is the shortest chromosome. 23rd pair is X and Y. So karyotype gives you a picture of all the chromosomes in the cells. And you can see bands on here, they're stained. And that makes the chromosomes visible. So karyotypes are done if they suspect a genetic disorder in the baby, and they can confirm if the baby has a disorder. So there's a few things that can happen that can cause disorders. Sure. So we can see chromosomes that are either missing or duplicated, or chromosomes that are just missing part of it, but not the whole thing that's missing. So on here, it's called a deletion, where part of the chromosome is just not there in the baby. Do you see which chromosome has been deleted? So it's a partial deletion. So it's going to be, part will be missing. So what we should see on here is that these homologous chromosomes are the same exact length. So if you see ones that are not the same length, then that's going to be deleted. So go through and see which ones are not the same length. Number 5, exactly. So number 5, do you see right here that this top part's gone? So each chromosome should be the same length about, and number 5 has a deletion right here. So that would cause a disorder. It's called cry-do-cat syndrome. It's French for cat's cry. Babies with this disorder, their vocal cords don't develop properly. They can't say words, and their cry sounds like a meow, like a cat crying. because their voice box is different and they're not able to vocalize like a baby would. Other times you can lose a whole chromosome. It's pretty rare to survive with a chromosome being lost, but there's really one case that is very common with this. Do you see what chromosome is gone?
Why?
The Y, yeah. So you see in here, this person only has one X chromosome and that's it. Do you remember what this is called? We talked about this the other day, when you only have one X chromosome and nothing else. Turner syndrome, very good. And this would be Turner syndrome, where this baby only has an X chromosome. And for females, they should have XX chromosomes, males should have XY. So it's the only chromosome you can really have missing and survive. And even in this situation, 98%, 99% of babies will die in the womb before they're born. So most babies do die from this before they're born. But the ones who do survive birth can live a normal life. But losing a chromosome is usually almost always fatal in a person. You can have extra chromosomes too, where you can have a whole or part of the chromosome that's duplicated. Do you see on here where that happened? Where do you see more than 2? Yeah. Okay. 21 exactly. So you know what this is called when you have 3 chromosomes in the 21? Down syndrome, exactly. So Down syndrome is a common genetic disorder. It's more common in older parents because older women, so the eggs have to undergo meiosis. Every month they undergo meiosis. One undergoes meiosis every month. But as the eggs get older, the chromosomes get sticky and they don't separate properly. And so it's more prone to have extra chromosomes in women who are in their 40s. And that creates Down syndrome. So Down syndrome can be a whole extra 21 chromosome, or it can be part of the extra chromosome. Either way, you get Down syndrome. All right, so that is the end of chapter 7.4. On page 205, work on those questions 1 through 7. Monday, we're going to do a lab, and then we'll start to review after that. So we're going to review on Tuesday and Wednesday. We'll have the test on Thursday next week.